Welcome to the chapter Unit 20 in the lab manual for Anatomy and Physiology in 1 lab. This unit is also called Unit 19 in the previous editions of the book. Now, in this chapter, we're going to be discussing the blood typing, components of the blood, basically how in the clinical medicine and the study of hematology, how do we deal with this tissue in the body. Keep in mind that blood is a type of connective tissue, just like cartilage, bone, and others. Now, normally when we take a patient's blood in the hospital, it's placed in the test tube, and that test tube taken to the lab and put in a centrifuge, which is essentially a large motor that spins the blood sample under very high speeds and essentially produces multiple different densities in the blood. They're separated in different layers based on density. And so the red blood cells, which is the heaviest component, drops down to the bottom. And then the rest, we have basically the plasma, which is mostly water, consisting of serum with their different components, and the white blood cells. So another way of looking at blood is basically we can say blood is separated into plasma and cells. Now, the types of cells in the blood are the white blood cells, red blood cells, and platelets. Now, first, let's talk about the basics. So, keep in mind we referred to blood plasma before. This is the extracellular matrix, the ECM component. In other tissues, in this case, this is the component that supports the cells of the blood. So, blood plasma is the watery component supporting the cells. When you separate the blood plasma further, you see that it actually consists of serum, which mostly is proteins such as albumin, which is a main carrier molecule that actually connects to hormones, drugs, medications, other things that you take into the body and allows those molecules to reach their target tissues, target organs. So again, albumin, an extracellular protein, it's one of the most important proteins in the body, is a carrier type of molecule and allows other molecules to be transported to different organs in the body through the blood. Other types of proteins, other molecules found in the serum, something called antibodies, which we're going to talk about soon when we talk about blood typing. And antibodies are basically very specific type of molecules generated by plasma cells in the blood that target specific antigens, and these are identifying molecules on top of the cell membrane of cells. Now, when we talk about white blood cells, WBCs, they're also called leukocytes. These are large cells, bigger than the red blood cell on a blood smear. They often have an unusually shaped, kind of multi-lobed, darkly stained nucleus. And there are many different varieties of leukocytes or white blood cells because they essentially deal with all kinds of problems in the body, such as part of the immune system, defending us against microbes, defending us against parasites, bacteria, viruses, and many other invaders or enemies coming into the body. In order for the many different types of white blood cells to know and be able to deal with many of these problems, we separate the white blood cells into these different types of cells that we find based on what kind of appearances they have, what kind of characteristics they have, and how they actually fight the infections. So there are two major categories. One is those types of white blood cells that have granules inside. These are pockets of chemicals that the cell uses to fight against things like bacteria and other infections. And if a cell does have these granules in its cytoplasm, we call these granulocytes. And there are those white blood cells that do not have these granules and use a different method of fighting against infections, and they are called agranulocytes, or basically cells with no granules. Let's talk about the granulocytes first. Again, these are large cells, really commonly found inside the blood smear, or basically in the blood when you see it under a microscope. Of these three, neutrophils, eosinophils, basophils, neutrophils are the most common, making up to 70% of all white blood cells. Neutrophils have granules. They're not often very easily discernible, but they are there. They're often very sort of light, kind of blue in color. And neutrophils are commonly associated with fighting against bacteria, other kind of general infections. For the purposes of this lecture, this discussion, we're going to focus on bacteria, and essentially say that neutrophils deals with common bacteria that might be entering the body, such as E. coli or strep, things like that. Again, neutrophils, very common, very important type of cell, one of the earliest cells to be found in the place of an acute infection, and this cell deals with common bacteria. Another important type of cell, but it's found in much smaller concentrations and numbers, is an eosinophil. Eosinophil is a cell that's found at around 5% at most of the population of other cells, and the eosinophil has very distinct red or pink large granules in the cytoplasm, and it uses these granules to fight against parasites and fungal infections, and allergies are dealt with as well. Obviously the allergies are not infections, but it's another immunological process that's very important as part of the ways that blood helps us in the body. So again, eosinophils will be dealing with things like parasites. Parasites are types of worms like hookworms, pinworm, tapeworm that you get from like undercooked meat or fish. Other infectious parasites deal with something like malaria or amoeba, like amoebiasis, schistosomiasis, and many others. Again, parasites are large organisms, often much more complex and bigger than bacteria or viruses, and need a special, different method to deal with them through the body. And again, the body usually uses these cells to fight against them. Basophils do a similar job. The only difference is that basophils are even in smaller percentage represented in the blood, at most only around 1%, or just a little bit higher sometimes. And these cells could be distinguished from eosinophils because basophils have really darkly deep blue or purple almost color granules that you can see under a microscope. And again, basophils will also be most likely dealing with either allergic responses or sometimes dealing with parasites or fungal infections as well. Again, very similar to eosinophils, but the cells will look quite a bit different under a microscope. Now, let's take a look at other cells that are also considered white blood cells. These cells now will not have granules that we could see. These cells fight using very different methods and are somewhat a little bit more complex, more often more long-lived cells. Two major categories are lymphocytes. So here we're thinking of the lymphatic system, lymph nodes and other lymphoid type tissues in the body, and monocytes. Now, lymphocytes is the biggest category. This involves cells like T lymphocytes, B lymphocytes, natural killer cells, and plasma cells. Of these four, the most separate sort of category is those of the plasma cells. These are the cells that actually generate the antibodies that many of these lymphocytes will use. Again, antibodies are specific types of molecules like immunoglobulin A, immunoglobulin G, so IgA, IgE, IgG, and others that will be used to fight against bacteria, viruses, and other kinds of organisms or any kind of invaders, enemies of the body, and including dealing with processes such as allergies and boosting of immunity during vaccinations and other immune-related processes. Now, general information about the lymphocytes, we can say that the T cells and B cells will normally deal with viral type infections, so viruses such as anything from a simple common cold type of virus to more complex viruses like the herpes virus or those related to different skin infections or infections involving pretty much any kind of organ in the body. So up to something like HIV even, or hepatitis C, hepatitis B, among others. Again, there are many different types of viruses. Keep in mind, viruses are a really unusual type of organism, essentially, one that shows no signs of life when it is outside of the host, outside of the cell, but then once injects itself into the cell, actually starts to reproduce, actually starts to bind its DNA into the DNA of the host and is able to replicate itself and show signs of life like a normal organism. Viruses could be enveloped, meaning they have a protein code that protects its internal genetic material, which also could be either DNA or an RNA molecule, right, depending on the type of viruses. Many different types of viruses exist, many that have nothing to do with the human body, those that attack only, let's say, plants or only bacteria or only other organisms. And there are many different ones that actually are pathogenic to humans, again, such as like herpes virus, for instance, or virus like HIV. The unusual type of cell here, also kind of a unique type, is the natural killer cell. This is a cell that, unlike other cells which use specific molecules to kind of find the antigens, find the identifying molecules on a cell that are being expressed and then attack those cells when they know that that's the enemy. For the natural killer cell, they actually are somehow able to find cells that do not properly express any identifying molecules but still able to target these for destruction. We do not yet know exactly how natural killer cells do that, but this is one of these kind of scenarios where this type of mechanism is used to identify and target specific cells. Quite often these would be tumor cells from some kind of a cancer, or cells infected by a virus that may be reproducing uncontrollably. Another type of cell without granules is a category of monocytes. These are commonly known as macrophages when they're found in the tissue. So when monocytes are actually just traveling throughout the body, these are white blood cells kind of looking for infections. Once the monocyte found infection, it kind of exits out from the blood vessel into the tissue. It is now called a macrophage. This is called from the word phagocytosis, which means eating or swallowing other cells. And that's basically what macrophages do. They're very non-discriminatory. They essentially can swallow anything from a deformed tumor cell to an infected cell to a bacterial cell to even kind of inanimate objects like a piece of sand or something that's potentially might be even harmless, but to a macrophage still is identified as something problematic because it doesn't belong in your body. So again, macrophages uses the process of swallowing other cells, or phagocytosis, to do that job. There's also site-specific macrophages or monocytes found in different organs, such as in the liver or in the brain, and those could be discussed separately. Now, for the other types of cells that are very important in the blood, we have obviously the red blood cells, RBCs. These are also called erythrocytes. These are the biconcave cells that are without a nucleus. These are one of the few cells in the body that are alive but do not have a nucleus inside of them. Remember, all of these cells originate in the bone marrow, deep inside bones, in that spongy bone component that we talked about. So when an early young red blood cell is first born, it's actually called a reticulocyte. At those early stages, it still retains its nucleus, and at that point, when it's in the bloodstream, it's kind of learning how to do its job. Once it has gained the ability to carry hemoglobin, that's the molecule that binds oxygen. Okay, it has that Fe or iron element inside the protein, and once the hemoglobin is able to be carried by the red blood cell, we now call this mature red blood cell, and this cell survives in the body for 120 days. After that point, basically the dying, essentially, red blood cell goes to the spleen to be sort of recycled, where we remove the hemoglobin, start to metabolize it, and the rest of the structure of the cell is discarded. Again, red blood cells, mature red blood cells, live in the body for only 120 days and are then kind of go through the spleen to be recycled, to be allow for essentially clearance of the cells and metabolism of hemoglobin. Remember, new red blood cells are always produced in the bone marrow, right? So we replenish those reserves. Again, most important function for red blood cells is the ability to carry oxygen throughout the body, right? To carry the oxygen to our cells, right? All cells need oxygen. Also, to some extent, hemoglobin is able to bind carbon dioxide. That's the waste gas that we exhale from the lungs. But primarily, hemoglobin is used to bind oxygen. Again, that is a reversible binding, right? So oxygen binds to hemoglobin just long enough to be carried to the tissues, to the cell, and then is dropped off so that the cells can actually pick up that oxygen. And the last type of cell, or actually cell fragment, something called platelets. This is where we remember that whenever we have bleeding or hemorrhage, when a blood vessel is cut or interrupted and blood loss is imminent, we have to stop that bleeding so the person doesn't bleed out, doesn't die. Right? So one of the main components of coagulation of the blood or blood clotting are these cell fragments which are very sticky. We call these platelets. There's hundreds of thousands of platelets per unit of blood. All the platelets aggregate and kind of bind together and stick to close off that open fragment in the blood vessel in order to stop the bleeding. Other components of coagulation are obviously different types of plasma proteins, different factors and others. But for the purposes of this discussion here, only focusing on the platelets. Again, they are cell fragments originating from the bone marrow that deal with coagulation of the blood. Now, after we talk about all of this, and essentially you look at the pictures in the book, look at the blood smears and the histology slides, the next important component in this chapter is to realize that sometimes when bleeding occurs, a person needs to receive a blood transfusion. Now, in order to receive a blood transfusion, we have to understand that there is a very specific way in medicine that we categorize the different blood types that exist. And based on these blood types and being able to identify the person with the correct blood type, match them to the correct blood that they need to be receiving, and kind of see how this works and what's involved in this important process in clinical medicine. Okay, so on the next page, let's take a look at the different blood types that exist. Now, in the United States, we use the ABO and RH blood system. In other countries, sometimes other systems are used as well. Here in the United States, we technically use others categories as well, but this is the most predominant, the most important kind of system used. Again, the ABO and RH system. Now, the most important thing to realize here is that when you're talking about a different blood type that a person has, whether you have heard about this or not, it's basically that you're visualizing your red blood cell and saying, on my red blood cell, I'm expressing some kind of molecules. These molecules that I'm expressing are called antigens. They're essentially like antennas sticking out of the house. They're sticking out on the top, the surface of the red blood cell. Whether you have a certain type of molecule expressed or not is based on essentially on what you've inherited from your parents. So we have no control of this, obviously. This is just essentially transcribed through our DNA. And we call these identifying molecules antigens. So for instance, if you are expressing A antigen on your red blood cell, your blood type is A. Right? You're expressing A antigen, your blood type is A. If another person is expressing B antigen on top of their red blood cells, their blood type is B. If you somehow inherited such antigens that you're actually not expressing either A or B, meaning again you have not inherited A or B antigens, your blood type is called O. And if you are actually expressing both A antigen and B antigens because you've inherited those from your parents, then your blood type is called or designated as AB blood type. Again, these antigens corresponding to the blood type, or the other way to say it is that to know what the blood type of the person is, you just look at what kind of antigens the red blood cells are expressing. Keep in mind these antigens do not change. They are produced by the body, by the cells throughout the person's life. You cannot modify these. It doesn't matter whether you have ever given someone blood or received blood from somebody. This stays intact forever and unchanged, just like your DNA stays the same in this regard. Now, another thing to consider here that's very important, especially when we talk about blood transfusion, is that our plasma cells in the body, those cells that generate those very specific antibodies we talked about, actually already know and produce specific antibodies within this framework. So for instance, for a person whose blood is blood type A, the body naturally generates antibodies that are anti-B. Now, you might say, why is it anti-B? The reason it's anti-B is, if you think about it, right, you cannot generate an antibody against your own type of antigen because you'd be destroying your own cells, right? That doesn't make sense. So the body kind of just in advance knowing that one day you might encounter a foreign blood, it will generate basically antibodies for the opposite type of antigen that exists. Okay, in this case, it would be anti-B. If another person has blood type B, right, remember this, that means they have B antigen. What kind of antibodies are they naturally producing? Their plasma cells are naturally producing anti-A antibodies. Our third person here who has blood type O, a very common blood type, this person, remember, doesn't have A or B antigens. Their body naturally, their plasma cells naturally produce anti-A and anti-B antibodies. And for the last person who is blood type AB, which means they have inherited both A and B antigens in their blood, the antibodies they produce would be, well, nothing. They don't produce antibodies within the system. Why don't they produce antibodies? It's because since they produce or express both of the markers or antigens on their cells, right, they cannot produce any antibodies within the system. Otherwise, they would be attacking their own cells. Okay. Now, just to make this just a little bit more complex, as we kind of often have to do in anatomy or in biology, there's that additional antigen system that we use. For the purposes of this discussion, to make this fairly more understandable and straightforward, I'm going to call this system as just Rh factor. In reality, there's actually multiple antigens involved here, but we're going to say either you have inherited the Rh factor, which means you get a plus next to your letter, or a minus if you have not inherited Rh. So again, if Rh is expressed on your red blood cell, we said there's a plus. If Rh is not expressed, meaning Rh antigen is not on your red blood cell, then you get a minus here. So when you combine this information with the other, you get this kind of a list of 8 different blood type possibilities. So these are 8 individual people, essentially, that could be there, right? So one person could have A positive blood, right? What does A positive means? They have A antigen and Rh. Another could be A negative. A means they have A antigen, no Rh. Someone else could be B positive, B negative, O positive, O negative, AB positive, AB negative. Again, if this is hard to understand at first, write them out, sort of make a table and try to write for yourself which one do you think has what kind of antigens and what kind of sort of parameters here are fulfilled. But basically, the most important to get from this is that when you are donating blood to somebody, you want to donate the blood, you want to make sure that the blood is going to the person who has either the same blood type as yours or only giving blood to a compatible recipient, right? So if you're in the hospital bleeding and need to receive blood due to your hemorrhage, you want to receive only the compatible blood. If you receive the wrong type of blood by mistake, you essentially will cause the system to be overwhelmed because the pre-made antibodies inside your blood will start to agglutinate or attack those foreign cells. It's like a rejection of an organ or a rejection of that foreign blood, and that causes dramatic overstimulation of the immune system, often attacking kidneys and other organs, causing multi-organ failure. This is what we call a transfusion reaction, right? It's always bad. Basically, there are many different types of transfusion reaction. One of the most important ones is TRALI, or transfusion-related acute lung injury, and there are many others. Basically, the point is here is that we want to always check the blood we're giving to the recipient, make sure it is the same type of blood exactly as the recipient, or at least the compatible type of blood. Now, this might seem really complex and sort of how are we actually going to deal with, we have so many different blood types and different people, but one kind of fairly easy way to standardize is to think about 2 main types of blood that we have of these 8 that kind of have these very unique features. So most of the blood banks in the United States and hospitals actually store a type of blood that is what's called universal donor blood. This is the blood that is safe to give to anybody. Now let's think about what does that mean, a blood type that's safe to give. This blood type has to be blood that when you give it to a patient as a transfusion, they will not react against blood. That means regarding what kind of antibodies the person will have, they will not react against that blood, meaning that blood has to have what? No antigens essentially in it, at least for this system. Now, what kind of blood would this be? This is universal donor blood. That's the O negative blood. That's the very common type of blood and the most important blood stored in blood banks. Again, think about O negative blood. What kind of antigens does a person with O negative blood has? They don't have A, they don't have B, and negative means no RH, right? So that's the safest type of blood to provide to a patient, even when you do not know exactly maybe what the blood type of the recipient is. And to turn this sort of upside down, in a different scenario, there might also be a person there who has a blood type where they can receive any blood type without fear of any interaction, of any blood transfusion reactions. This is a person that's a universal recipient, that's what we call them. This is the AB positive blood. If you have AB positive blood, you have A antigen, B antigen, and RH antigen there. And that means, what kind of antibodies do you actually have in your blood? None. You don't have any antibodies. Since you don't have any antibodies, that means regardless of what kind of blood you receive, you will not react against that blood. Now, O negative is universal donor blood, AB positive is universal recipient blood. Keep this in mind. Read this chapter, look over the notes, look at the material there. In the homework, they will ask you different questions, different ways to kind of identify what kind of blood is compatible to which patient. This information, again, I recommend after you watch the lecture video, read the chapter, do the homework there, and again, pay attention to the specific varieties of these different blood types and being able to compare the antigens and antibodies and what kind of possibilities you get. And obviously in the beginning of the chapter, make sure to review the components of blood, the white blood cells, red blood cells, platelets, and other components in the serum.